Hello and welcome to STEM with Mr. N, where I perform different demonstrations and explain the science behind what we're seeing. This week I'm going to show you how to extract DNA from strawberries. Let's check it out. All living organisms contain DNA. DNA, which stands for deoxyribonucleic acid, contains the blueprint for an organism. Overall, it tells the organism how to grow and function. I've done a video previously on DNA looking more in depth at it and its structure, so I'll put a link in the description for you to check that out. But have you ever wondered how scientists can extract DNA from plants, animals and humans? Well, today I'm going to show you how to make a DNA extraction kit and how to extract DNA from strawberries. For this activity, you will require a tall glass, a funnel, a smaller jar, some dishwashing liquid, some water, bamboo skewers, 3 strawberries with the leaves removed, a sealable freezer bag, some salt, some cheesecloth, some measuring cups and measuring spoons, a small bowl and spoon for mixing, and some rubbing alcohol, or in my case I'm using vodka. Because this activity involves using alcohol, this should not be done without an adult's permission and without an adult present. The first thing I'm going to do is mix 1/3 cup of water with half a teaspoon of salt and a tablespoon of dishwashing liquid in my small bowl, mix it together and then set it to the side for just now. Next, I'm going to completely line the inside of my funnel with cheesecloth and then put this inside the tall glass and set the glass to the side for just now. Next, I'm going to put the 3 strawberries into the resealable freezer bag, push out the excess air and seal the bag tightly. I'm then going to use my fingers to squeeze and smash the strawberries inside the bag for around 2 minutes. Now that the 2 minutes is up and the strawberries have been squeezed and squashed, I'm going to add 3 tablespoons of the water, salt and dishwashing liquid mixture that I put together earlier. This is my extraction fluid. Once I've put the 3 tablespoons in, I'm then going to press out the excess air and reseal the bag again and squeeze everything together for another minute. Now that minute's up, my strawberry mixture is starting to look quite frothy because of the dishwashing liquid that's just been added to it. I'm going to open this bag and pour the mixture into the cheesecloth. I'm going to let it drip until there is nothing but wet pulp left in the cheesecloth. It can take a while for the liquid to run through the cheesecloth and be left with that wet strawberry pulp on the top. So set the glass and the cheesecloth to the side, put the rubbing alcohol or vodka into the freezer to cool it down, and come back to your glass in about 10 to 15 minutes time. Once you're happy that the liquid has run through and you're pretty much just left with strawberry pulp, measure out half a cup of the cold alcohol. Now that I've got my liquid that's run through the cheesecloth, it's time to add some of this into a small jar. Spice jars and baby food jars are good sizes to use. I don't have either of those, so I'm using slightly smaller jars. I'm also going to use a small funnel so that I don't spill the liquid all over the table. I'm going to pour some liquid into each jar until the jar is about 1/4 full. Then I'm going to very carefully pour some of the cold alcohol down the inside edge of the jar, and I want to make sure that I don't pour it in quickly. I just want to trickle it down so that the 2 liquids don't mix until I've got about 1 inch of alcohol sitting on top of the strawberry mixture. Now that the alcohol has been added to my jars and to my glass, I just want to sit back and observe what is happening for a couple of minutes. By looking closely, I can see that there are white clumps forming on top of the red liquid and starting to spread up through the alcohol layer. This is the DNA from inside the strawberry that has been separated out, and it is starting to rise up into the alcohol layer. So what is actually happening in this activity? Well, when I added the extraction liquid, that was the dishwashing liquid, water and salt, into the crushed up strawberries, the dishwashing liquid made the cells of the strawberries lyse. That means they popped open and it released their DNA into the bag. The salt created an environment that would allow the DNA to clump together. When I then added the cold alcohol to the strawberry liquid, this allowed the DNA to rise away from the rest of the strawberry, and that is why we saw those white clumps of DNA inside the jars and inside the glass. So why do scientists need to be able to extract DNA? Well, scientists extract DNA for all sorts of different reasons. DNA can teach scientists about how the human body functions, help them make new medicines, help them catch the suspects in a crime, and we can even learn about people that died thousands of years ago by extracting DNA from their bones. Scientists go through a similar process to what I have done in this video by using a chemical agent to lyse the cells, that means pop them open and release the DNA, and then using another chemical to allow the DNA to extract itself from the rest of the cell, and the scientists can then gather this up and examine it. Well, that's all for this week. I hope you enjoyed it. If you did, please hit the like and subscribe button so you can stay up to date with all future STEM with Mr. N videos. As always, I would like to take this opportunity to answer any science questions you have about any science topics at all. So feel free to email me at stemwithmrn@outlook.com and I'll get back to you with answers to your questions. You can subscribe to the channel by pushing the button here, and I've added links here to the other STEM demos I've done so far, here to my STEM career interviews, and here to my robot review videos. This has been STEM with Mr. N, extracting DNA from strawberries.